Hello viewers, now we are going to discuss about classification of enzymes. Enzymes can be classified on various bases and we are going to begin the classification based on the site of action. On the site of action basis, how enzymes are classified, we are going to discuss. Enzymes are classified into two groups named endoenzymes are also called as intracellular enzymes and second one exoenzymes also called as extracellular enzymes. So what are endoenzymes? These enzymes acts inside, endo meaning inside the cell, also called as intracellular enzymes. These enzymes are responsible for synthesis of cell components, production of energy and metabolism. In all these process, endoenzymes plays a very important role. Examples of this enzyme is synthetase, isomerase and phosphorylase. So which are the enzymes which are produced and acting inside the cell, they are called as endoenzymes or intracellular enzymes. Most of the metabolic pathways which takes place inside the cell are performed by these endoenzymes. Maybe like glycolytic enzymes, Krebs cycle enzymes, they are all endoenzymes because their reaction takes place inside the cell. Exoenzymes, exo means outside, so these enzymes are acting outside the cell. Even though they are produced inside, they are acting outside the cell. They act outside the cell and are called as extracellular enzymes. Such enzymes are digestive in their function. Most of the digestive enzymes belongs to this category. It helps in the breakdown complex molecules into small units. Example: proteases, lipases, and amylases, which are involved in digestion of respective biomolecules, proteins, lipids, and carbohydrates. In this illustration, you can see the two cells. In the first cell, you can see the enzyme in blue color, and they are combining with the substrate outside the cell, which is in red color. Substrate and blue are combining, and active enzymes are present outside. They are exoenzymes or extracellular enzymes. In the right side, you can see yellow color enzymes which are present inside the cell. There also they are combining with substrate to form product. Since these enzymes are within the cell, they are called as endoenzymes or intracellular enzymes. This classification is basically on the basis of location of active enzymes, whether they are outside exoenzymes or whether they are inside endoenzymes. Now the very important method of classification of enzymes, and this classification is based on the type of reaction catalyzed by the enzyme. There are six classes of enzymes which are classified based on the reaction they are going to catalyze, and they are in the left side. You can see six classes are there: oxidoreductases, transferases, hydrolases. Fourth one is isomerases, lyases, and ligases. These six important classes of enzymes are going to perform various biochemical reactions. So let us see one by one in detail. So oxidative, you can see transfer of hydrogen and oxygen atoms, and examples in the last column you can see dehydrogenase and oxidases. Transferases involved in the transfer of specific group, example transaminases and kinases. Hydrolases helps in the hydrolysis with the help of water, example esterases or digestive enzymes. Isomerases helps in the change in the molecular formula from one form to another, example phosphoxoisomerase, and so on. Lysis helps in the breakdown of substrate into product, or removal of a group or addition of a group into a substrate, example decarboxylases. Ligases means synthetases, means joining of two molecules by formation of new bond, example citric acid synthetase. So this I am going to explain in detail. So let us see enzyme classification. Enzymes are classified into six classes based on the type of reaction catalyzed. The first one, as we have discussed, oxidoreductases. In this, we are going to see in detail of each of the enzymes. Oxido means oxidation, reducto means reduction. Oxidation and reduction, two reactions is performed by this group of enzymes, and oxidoreductase class of enzyme catalyze the oxidation and reduction type of reaction. You can see in this AH and plus B, arrow mark A plus BH2. What is oxidation? Oxidation means removal of hydrogen. And what is reduction? Reduction means addition of hydrogen. Removal of hydrogen is oxidation, addition of hydrogen is reduction. So if in this figure, if you see AH is there, plus B. So in the left side, A is having H and B is not having H. So after the arrow mark, you can see the A plus BH2. That means from the A, H is removed. That means A is undergoing oxidation. For B, H is added. That means B is undergoing reduction. This reaction is performed by the oxidoreductase enzyme. This is illustration, and specific example is given below. Glycerol 3 phosphate converted to 1,3 bisphosphoglycerate. In this, NAD is converted to NADH2. If you consider glycerol 3 phosphate as AH2 and NAD as B, so NAD is ending with only D, not H before arrow mark, and after the arrow mark you can see NAD is having H after the arrow mark. So that means it is very much similar to the above illustration B, where B is not having H in the beginning and then later it got H. So glycerol D 3 phosphate, by losing the H, it is converted to 1,3 bisphosphoglycerate in the right side after the arrow mark. This reaction is performed by a enzyme called as glycerol 3 phosphate dehydrogenase. D means delete first two letters De. Hydrogenase means hydrogen is removed, delete the hydrogen. From which one hydrogen is removed? From glycerol 3 phosphate hydrogen is removed. That's why the enzyme name is glycerol 3 phosphate dehydrogenase. It belongs to the oxidoreductases category, and few more examples for this category are lactate dehydrogenase and pyruvate dehydrogenase. In the ending you can see the other examples. So this is about the first class of enzyme, that is oxidoreductases. Next we'll go for transferases. Transferases catalyze the transfer of a group from one substrate to another substrate. So transfer of a group, chemical group, okay, not hydrogen. If it is hydrogen is involved, it is called as oxidoreductases. Other than the hydrogen, like chemical groups, aldehyde, keto, amino group, then such type of group is transferred from one substrate to another. Such reaction is performed by transferases. Transferases catalyze the transfer of a uh, group from one substrate to another substrate. In this illustration, you can see A with X and plus B. If A is attached to X, which can be any group, the X may be aldehyde, keto, amino, any one of the chemical group. After the arrow mark, you can see A is free and B is got the group which was previously present with A. So B got the group. If aldehyde is the group, if you consider as X, which is present with the A before the reaction, after the reaction that aldehyde is transferred to the B substrate. So this type of transfer of chemical group is performed by transferase enzyme. And a specific example, if you want, glucose arrow mark glucose 6 phosphate, ATP is converted to ADP. If you consider glucose as B, if glucose is considered as B, ATP as A. So ATP means adenosine triphosphate, tri means 3 phosphate, it is converted to ADP. That means adenosine diphosphate, only 2 phosphates are there. The one of the phosphate from the ATP is transferred to glucose, and it is attached to the sixth carbon of the glucose. Only one phosphate is transferred to glucose, that is attached to the sixth carbon of the glucose. That's why it is called as glucose 6 phosphate. And the reaction is performed by glucokinase. These steps comes in glycolysis and other glycogen metabolism pathways. And glucokinase is a transferase enzyme which is involved in the transfer of phosphate group. The other example for this group is phosphoroctokinase and transaminase. Transaminase, as the name itself indicates, transfer of amino group. Transaldolase means transfer of aldehyde group. Transketolase means transfer of keto group. Transesterase means transfer of ester group. Like this, so many transferases category enzymes are available based on the type of group is transferred. So this is second category enzyme. Next one we are going to talk about hydrolases. Class 3 enzyme, that is hydrolase. Hydro means water. As you can see in the first sentence, hydrolases catalyze the splitting of molecules. Splitting means breakdown of molecules by hydrolysis. Hydrolysis means, in bracket you can see, by using the water. In this illustration you can see just AB, it's a substrate. It is combining with the water H2O, and enzyme is involved in this reaction. The enzyme is going to take the water and break it into H and OH, and H is going to attach to A. After the arrow mark you can see H is attached to A and OH part is attached to the B. So AB, which was attached or is in a single molecule in before reaction, you can see AB. After the reaction, that means after the arrow mark, you can see A is separated from the B. So A and B, two products are obtained by the breakdown of this hydrolase enzyme using water molecule. If you want specific enzyme, you can come across lactose plus water. Lactose is AB if you consider, and then H2O. Very much above similar illustration. The specific example you please follow it. And above the arrow mark you can see lactase, that is the hydrolase category enzyme. After the reaction, as you can see above, AH plus BOH, A and B is separated, and you can see in this same manner glucose and galactose is separated by the lactase enzyme using water. So lactose, which is broken down into glucose and galactose by lactase in presence of water, is the specific example. Along with that, if you want other example, you can see in the bottom maltase, sucrase, etc. These are carbohydrate digesting enzymes. Similarly, you can come across protein digesting and lipid digesting enzymes, which also comes under hydrolase category enzymes. Next we will go for fourth category enzymes, that is lyases. Lyases catalyzes the splitting of molecule by mechanism other than hydrolysis. Lyase, as the name itself you can see in the lysis, you can see the in the paragraph last word hydrolysis. Lysis or lyases, there is a similarity. Lyase or lysis means breakdown. Lyases catalyze the splitting of molecule by mechanism other than hydrolysis. Other than hydrolysis means in this case there is no addition of water. In the general illustration you can see AB, which is a substrate, is going to combine with certain molecules in presence of enzyme, and it is going to separate into A plus B after the arrow mark. So this is about AB getting converted to A plus B without water. In this case you cannot see any water. And in the specific example, if we can come across fructose 1,6 bisphosphate, it is converted to glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, and it is performed by aldolase enzyme. So aldolase is going to convert the fructose 1,6 bisphosphate to two molecules. In this case there is no addition of water. And similar examples, if you want to come across fumarase and aldolase B, is the example which comes under this lyases category. In this you can just consider breakdown of substrate into two products but without water. Isomerases. So isomerases means iso means same. Molecules having same molecular formula but different structure formula, they are called as isomers. Isomerase class of enzyme catalyze the interconversion of isomers. For example, if you see the illustration in this A arrow mark A dash, or instead of A dash if you write small letter A, that is very convenient way of explaining. For example, A arrow mark, you consider it not capital A, small a. So what capital A converted to small a, for example, that means what is the meaning of this A? A is A only, but we only it is getting converted to capital A to small a. That means structure is changed, and that's why This type of interconversion is called as isomerase. Glucose 6-phosphate is converted to fructose 6-phosphate and it is performed by isomerase enzyme. If you see before arrow mark and after arrow mark, glucose 6-phosphate and fructose 6-phosphate both contain phosphate. That's why the enzyme name PHOSPO above the arrow mark, and both glucose and fructose contain 6 carbon. That's why exose, and both are isomeric forms. That's why enzyme reaction and enzyme is isomerase. So this comes in glycolysis. Glucose 6-phosphate is converted to fructose 6-phosphate and both are isomers having same molecular formula but different structural formula. So similar to this type of enzymes, examples are phosphotrioisomerase and epimerase. So this is about isomerase enzyme, which is class 5. Next one is we are going to discuss about is the last category of enzyme, that is ligase. Ligate means join. It's a English meaning. Ligate means join. Ligases catalyzes the joining of two compounds. The energy of ligation is obtained from by ATP hydrolysis. In this enzyme, two substrate, for example, if in the illustration if you see A plus B, two separate A plus B is going to combine and are joined to form a single compound or single product called as AB. Whenever you want to join two molecules, you require energy, and energy is derived from ATP. ATP is converted to ADP for binding A with the B. This is illustration. Specific example if you want, you can come across acetyl CoA plus carbon dioxide. If acetyl CoA is A and carbon dioxide is B, it is going to get converted to malonyl CoA, which is the AB. And in this reaction also, in the below the arrow mark, you can see ATP converted to ADP. For joining the two compounds, energy is required in presence of manganese and biotin. These are cofactors and coenzymes, and they are catalyzed by acetyl CoA carboxylase enzyme. Since carbon dioxide before the arrow mark you can see is utilized for the reaction, that's why the enzyme name is carboxylase, and that carbon dioxide is attached to acetyl CoA. That's why the enzyme name is acetyl CoA carboxylase. So enzymes names itself will suggest lot of meaning and information. So you have to be uh, consider those points regarding a reaction also, and the name of the enzyme. Is there any linked naming is there? So next is in this category certain other enzymes are glutamine synthetase, pyruvate carboxylase, etc. This you can come across the mnemonics. In the last you can see, oh thank heaven, love is life. So it is some mnemonics. That means you can see the first letter of each of the word O, that is in bold letter OTHLIL. It indicates about O means oxidatases, T means transferases, H means hydrolases, L means Lyases, I means isomerases, ISO isomerases, Li means LIGASES ligases. So if you want to remember this in easiest way, you can use this mnemonics for remembering the enzymes name. We'll continue the enzymes in the next session.